So welcome to this week's episode of Leaders on a Mission, where I'm joined by inspiring leaders driven by the impact of creating a healthy and sustainable world. Now, in this week's episode, I'm joined by Joel Stone, President of Convergence Advisors and Senior Vice President of Strategy for Lee Enterprises Consulting. Joel is a respected leader within the industrial biotechnology field, and his job is all around helping companies take the promise of science and helping scale and commercialize it. So I've got to know Joel pretty well over the last few years, and it's great to have you on the show. Thanks very much for joining me, Joel.
Well, good morning, Simon, and pleased to be on as well. I've watched a lot of your other podcasts with various people, and it's amazing how many people you've had on that I know that are, and many of them work within the same space, and we all know each other. So it tends to be a small pond when you're looking at industrial biotech and synthetic biology. So again, so pleased to be on and spend some time with you today.
No, that's great. That's great. And it is a small, and it's a very niche market, but I think the interesting thing about the marketplace is it does cut across a number of different industries. And that's one of the things I've always loved about it, really, is there's this divergence with other industries, but I think when you come back to your network, it's amazing, as you've said, how many people we do have in common. So yeah, absolutely. So maybe I'll take it all the way back, Joel, and think back to your kind of the early influences on a young kind of Joel Stone kind of growing up, really.
Oh, certainly. No, it's interesting path I've taken, but I guess one of my most fortunate times was when I was, believe it or not, when I was in junior high school. I spent a summer, with my uncle, who was a PhD organic chemist and PhD physicist, and he actually taught at the University of Alabama. And this is a true story. I went down to spend the summer. I had two cousins that were about the same age. And when I was down visiting with him, I actually went to his organic chemistry classes. When I was a junior, I loved chemistry. I just loved the sciences. And he thought it was nuts, saying, "Hey, Uncle Fred, can I come to the classes and listen in?" Well, which I did. And then he ultimately went on to become the director of the National Science Foundation here in the U.S., which is quite an esteemed position to be in, because it basically manages all the early science programs. So that got me exposed even more early on with some of the things that were being done at the NSF in this whole area of biotechnology, sort of fitting in to work hand in hand with chemistry. And that was 40 plus years ago. So, back in that day, people didn't even know what enzymes were. It was just there are these biocatalysts that seem to be able to do some unique things. So, I got sort of entrained into that early on. And actually, when I was graduating from high school, I actually sought out some of the universities that not only had a chemical engineering department, but were leaning towards biochemical kinds of engineering. And ultimately at Virginia Tech, we had a couple professors that were brought on that were biochemical oriented. So I was on the leading edge at Virginia Tech on developing a biochemical engineering department. So two just sort of lucks of the draw, so to speak, but very good mentors, getting me figuring out what I want to do when I grow up. And it got me thinking about what I want to do is build things at a large scale. I didn't want to do R&D. I wanted to be a commercialization kind of person. I think I got that from my dad, because he worked for General Motors and was in charge of body moldings and parts at one of the General Motors plants early on. So he was into manufacturing, and I got that gene from him.
Great. No, that's great. And you spent your whole career in the space, right? And
yeah. I've spent my whole career in industrial biotechnology, and now drifting more and more towards synthetic biology. But my expertise is large scale fermentations, whether it be for fuels or chemicals or foods, across the entire gamut. Early career, believe it or not, I actually started out working at Eli Lilly, which was pretty fortunate. My first job right out of grad school. But strangely enough, at Eli Lilly, even though my background was heavy in biotech, the massive project that they had going on was actually an organosynthesis project to make very strong herbicides, and within one of their subgroups. So that got me exposed to some of the strange chemistry side of things, but soon moved back over into the fermentation area.
Yeah, no, great. That's great. And tell me, I suppose when you look through your career then, when you look back, maybe outline a couple of projects or positions that you've worked on that, where you really reflect back fondly on.
Well, I was, early on, I took a position at A.E. Staley, which today is known as Tate and Lyle. And my assignment was to build the first ethanol plant that Tate and Lyle was going to build. And that was in Loudon, Tennessee. The plant still runs today. It's a massive, biochemical complex, converting corn into all different kinds of products. Genomatica actually has did a joint deal with DuPont building a facility there, more recently. But I was actually in charge of building the first ethanol plant. It was one of the first large scale plants here in the US. That was back in 1982, when none of the other biofuels industry was even around. This was one of the first plants built in the US of any size. And I ran the project, and then I actually volunteered, to move out of engineering over into operations, and actually went down, and what I designed, if it wasn't going to work, there was only one person to blame, and that was look at myself and go, what did I do this for? But it was a very successful startup. And just think of it, in 1982, this was a plant that was a full wet mill, ethanol facility, made corn syrups, et cetera, full rail yard, wastewater treatment. In 1982, a $310 million plant.
Wow.
Just convert that into what it would cost in today's dollar. So that was the magnitude of the project. But really a great team of people. It was a team-based management system. Everybody in the plant was on salary. There were no hourly employees. So it was not only a first ever, technological feat to build this plant, but also a giant management experiment that was also very successful. That really got my career off. And I was in my late 20s managing a project of that magnitude. So it was pretty incredible.
Yeah. How long did it take to kind of commercialize or to complete the building?
That was a 2-year project. As far as the construction part of it, probably about 2.5 years in total, which was still, for that size of facility, a lot going on, a lot of moving parts. But we had an outstanding management team across all boards. Nearly every one of us have got from that facility. We all stay in contact with each other, and we've all moved on to be probably, I would say, very successful in the different areas that we've gone into. Some in especially chemicals, some stayed in fermentation like myself. We had expertise in various types of unique extraction processes. So in fact, I was just talking to the fellow who was the technical manager during that project. He now lives in Portugal. I was catching up with him a couple of weeks ago, asking him, hey, what is it like to live in Portugal if I suddenly wanted to bail out of the US and move to a Euro country?
Beautiful. I bet he told you it was beautiful.
Oh yeah. He showed me, turn this camera around, he's right near the coast, so you can see the Atlantic Ocean from his back porch. So I'm going, oh, not bad. Got the nice cool breezes, don't need air conditioning.
Absolutely. No, it's beautiful. And but I would imagine since 1982 or since that project, I mean, the journey of the industrial biotech sector has been an interesting one, right? And I suppose when you survey back and you think of where we are now and you think back then, what have been the biggest changes, do you think, in terms of, the sector and how they've evolved?
I would say the technology, the measurement technology and instrumentation controls, especially all the things that you can do around a large scale fermentation to be monitoring oxygen uptake rates and cell densities and agitation inputs. Even the changes that have been remarkable on the reliability of pH probes. Measuring pH in a fermentation is pretty critical, and temperature controls, dissolved oxygen rates, all that kind of stuff. Then I would say that's probably in the early stages the most remarkable things, and the ability to better design bioreactors in terms of the type of agitation. A lot of technological advancements in turbine designs for delivering the right type of agitation within a fermentation, and then cranking up to more recent times, which is what's really creating a whole new renaissance of sort of reinventing the chemical industry, reinventing the food industry, in fact, and that is the onset of synthetic biology, combining engineering, microbiology, artificial intelligence, mathematics, and algorithms, all those things converging to be able to predictably figure out what changes to be made in an organism, to have it create something down a unique metabolic pathway. And all this work that's been done in the synthetic biology space, the culmination of that, so to speak, a lot's going on. I follow it mostly on the industrial chemicals and food side of things, but all this work being done with messenger RNA, with being able to have a COVID vaccine developed in less than a year, is all about all these things that can be predictably determined using synthetic biology. So it's just amazing. 20 years ago or even 5 years ago, it would take 5 to 10 years to develop a vaccine, and here we developed one very unique for a problematic virus in less than a year. It's just incredible. It's like Moore's law applied to biology, the rate that we're able to make improvements. And now the things that we're looking at to develop the foods of the food protein, a big area now. One of the two largest problems we face as humanity, well, one is climate change, that is reducing CO2 concentration, and the other is feeding people. It just so happens both of those are converging for a train wreck by 2050 unless we do something. And in many respects, Simon, they actually go hand in hand, because there's so much that's done with the agriculture, with raising cattle and swine and poultry, that if that can suddenly be done by agricultural means to produce agricultural proteins, but my focus is on producing some of these proteins and flavors, carbohydrates via fermentation. It's not a dream anymore, it's reality. Some of the things that are going on at the bench scale and now starting to move into larger scale. So for someone like me that started out as a early biochemical engineer, studying under the guys that wrote the textbooks, back 40 plus years ago, it's pretty exciting to see what's happening now. See all these things that we've been dreaming about for years and years and years is becoming reality. Displacing normal fossil-based chemicals with bio-based. Pretty incredible stuff.
Absolutely, and it really is a very exciting time in our history, and the potential is enormous. Just a question for you. when you consider about, you speak about the synthetic biology, you speak about the organism design side of things where there's been so much kind of progression with. One thing that organizations continue to struggle with, because I hear them, and that is the scale up, for instance. The scale up of the organisms into a commercial kind of viable, real kind of business, as it were. And I'm wondering, what are the fundamental kind of challenges where getting something in a lab, getting it working, and then being able to then create a business of great scale?
The biggest challenge is, and the way I look at it, the way I explain to people is I look at scaling down. I don't look at scaling up. So, where I found my strength to be through the years is I envision what the large scale plant is going to look like or should look like. How large should it really be, or how large does it need to be? Because bigger isn't always better. Sometimes there's mechanical limitations, sometimes there's mass transfer limitations, sometimes there's shear limitations on the type of organism that you're dealing with. Some fermentations are much more susceptible to contamination because the organism isn't robust. So there can be a small amount of contamination that suddenly takes over. So those are all the things I think about. And then I start defining what are all the parameters that I have to better understand at a small scale, and which ones can be sort of screened and understood better at the bench scale, which ones need to be studied much more at a pilot scale. And then quite often people will try to go from bench to pilot and go from pilot directly into a commercial scale. Sometimes that can be done if you really have your I's dotted and T's crossed, but more often than not, there's a higher failure mode because the rate of scale, you're limited as far as really reasonably how much what range of size changes you're actually making from that pilot to the commercial. So quite often I recommend for people to go at a demonstration scale, sort of an in-between pilot and large scale. And quite often in doing so, in most businesses, when you're looking at the business development side and, how is the product going to be used, the quality characteristics that the customer wants, it's quite often best to make demonstration scale quantities that you can move into your ultimate customers for them to do formulation testing or application testing, going into their final branded product or industrial type of product. So I tend to, and demonstration scale is also quite important on the biotech side, because that's where you really get to test out the DSP, or what we call the downstream processing. Because downstream processing isn't always straightforward. And what I found was when you go to larger scale fermentations, you might see other components that the organism is producing that would be impurities or would be things that you don't want the finished product. And quite often you don't see those at the bench scale. You don't see them until you get the pilot, or often when you get the demonstration scale. And then you've got to be figuring around with the DSP. If there's a particular contaminant that's in part per million levels, it might give an off color or an off flavor. How do you remove that? What downstream process? So there's where you reach the intersection of, the biochemical or fermentation engineers focused on the fermentation and the microbiologists, but you now need really good chemistry and quality assurance types of people, and good downstream processing, which is basic blocking and tackling of chemical engineers. You don't have to have a biochemical engineer that's determining which membrane system you're going to use, or what kind of centrifuge you're going to use, or crystallization or drying. That's all basic chemical engineering blocking and tackling. And the technology is just, circling back to your earlier question, the technology for downstream processing and chemical processes has also gone by giant leaps through the years. As an example, I'm working with a client company right now that's developed a very unique nanofiltration membrane. And why it's unique is it's non-fouling. Trying to find membrane systems that don't foul when you're dealing with sticky proteins and lipids that want to coat out on surfaces, all of these different kind of, molecules that want to foul and coat out on surfaces. Well, this is a technology they've developed that I've never, in my entire career, I've never seen anything like it. It can handle some of the nastiest streams that I've ever confronted, and it doesn't foul within 24 hours. It can run for days, and then it can, with a gentle wash and very mild chemical, treatment, completely recovers to 100% flux rates. It's just miraculous. So those are the kind of things that, modern day chemistry and, being able to work through very complex engineering and chemistry and microbiological issues all together, that we're able to do things that, we couldn't do 5 or 10 years ago. And that to me, even at my gruffy age, is pretty exciting to see that kind of stuff being developed and coming out of the universities and our young scientists.
That is great. And what about when you think about innovation and you think about some of the challenges that you've mentioned with scale up, for instance? Is there? So I'm not thinking more about the downstream process, but I'm thinking about the fermentation kind of scale up. Are there kind of innovations happening now or in the future you see, which will make that journey of scale easier and more predictable, and there'll be less risk associated with that, for instance?
Yeah, I think a lot of it revolves around sensor technology, being able to measure what's happening real-time basis in the fermenter. A lot of that's being done at really small scale. I've seen these small 5-liter fermenters that they look like a porcupine. They have so many sensors on them that are measuring anything and everything that's going on. To measure the metabolism of the organism, the cell density within the fermentation medium. I wouldn't be surprised if before long there's sensor technology that can tell you if a cell is looking abnormal. If it's sort of getting discombobulated because it's not getting the right amount of micronutrients. And then I think, just like any living organism, all of us, whether it's a human being or farm animals, all the way down to bacteria and fungi, there's certain micronutrients that they need or should have that allow them to thrive. It's like giving the organisms the right types of vitamins and minerals and peptides that allow them to be healthy. And I think there's a lot more being developed and understood around that, because that's what allows you in a industrial fermentation to maximize the yield and do it cost effectively. Not having to overdose a certain type of nutrient. Mm-hmm. And lots of times when you move from the bench, at the bench scale, quite often you're using the sort of standard modelized nutrient packets that are sort of a standard that's been used for certain types of organisms, that quite often, if you try to use those same materials at a large scale fermentation, you couldn't do it cost effectively. So you've got to look for nutrients and additives that can be procured at a large scale, to allow you to do those fermentations cost efficiently. And I think there's a lot more work that's starting to go on doing that, in today's world, with the microbiologists, and now having the microbiologists work hand in hand with a with a biochemical engineer or fermentation engineer that understands the sensor technologies, understands that, even though in theory you think you could produce something in a 500,000 liter fermenter, it may not make mechanical sense or operational sense to doing that. So scale it down and do multiples in 150,000 liter fermenter. That if you have a problem, if you have a contamination, if you have a power outage and lose air, and you've got to throw a fermenter away, it's much easier to throw away 100,000 liters worth of fermentation broth than it is 500,000 liters. Let me tell you, I've been there. I've seen it, and it's all oriented towards the type of fermentation too, that you're trying to achieve, and the quality characteristics, of course.
Yeah, absolutely. Who needs a power outage on a 500 million liter fermentation? Yeah, but do you see these happening, like random acts of kind of power outages and things like that?
When in most plants, from an engineering standpoint, on large scale facilities, pretty much the standard when you're building a large facility is you'd have power coming in from two different sources. So if you have a lightning strike or a bunch of birds up on a wire that something breaks, you got a power coming in from an alternative source. And in some cases, if facilities put in, for critical kinds of equipment, they put in emergency generators that can provide power in the event of a power outage. So many of the facilities I built in my earlier career producing enzymes, we had backup power, and we had power looped in from two different locations. So it was very seldom that we'd ever have any kind of power outage. That'd be something really quite unusual for that to occur.
Yeah, I got it. And tell me, if you consider startups now and you think about organizations that are scaling, there's a manufacturing or commercialization strategy each company needs, as it were. And there seems to be a real kind of emphasis for companies not investing in building their own manufacturing facilities over the last 10 years or so. Is this a trend that you continue to see, or do you see it kind of firms, wanting now to, to spend the kind of CapEx on building their own facility?
It's actually, Simon, that's a great question. It comes up quite often. There's not an either or is my direct answer. Certainly there is a preference to not spend a lot of CapEx, just the investor appetite in all these startup companies that have venture investment. Venture investors don't like to put money into bricks and mortar. The problem we're running into is there's a significant shortage of contract manufacturing capacity. So it's very difficult to take the model that the Apples and Dells, and the Samsungs, all these companies who don't own any of their own manufacturing. They source all the parts and have them produced at a contract facility. Foxconn as an example for Apple. The capital investment, yes, it's high in those facilities for building cell phones and all kind of electronic boards and gadgets and things that go into automobiles and appliances. Building a contract manufacturing to do fermentation is quite an undertaking, or even building your own plant. It's very capital intensive. And even spending 2 years in looking at, what a manufacturing facility would look like. I've got firsthand experience because, interviewed over 150 different synthetic biology, biotech companies that were looking for manufacturing capacity. So it was my task to try to figure out what should I build that could meet the needs of as many of those companies as possible. And there were, aeration rates, agitation rates, the DSP were all aspects. But the key one was, how small do I want to have fermentation capability? But more importantly, what's the largest size fermenter that I might want to have that would have the utility of servicing multiple different clients within the next 5 years or 10 years. And it was a very interesting undertaking. We ended up not building the facility because it was going to be so capital intensive that we couldn't find investors with the right terms. Now that's changing now. I mean, I think in when I was working on Fermentum, we were a little bit ahead of the curve still, in terms of the real need for contract fermentation. That's really relates to here in North America. There's quite a bit of contract fermentation capacity in the EU because it's been supported and subsidized by the government. There is being supported by the DOE, what's called the Manufacturing Innovation Center that has just recently been funded, which is going to be focused on industrial biotechnology, scale up and manufacturing. And it's actually based in Minneapolis, Minnesota, but they're going to build out facilities throughout the idea is to build out facilities for scaling throughout the US. And I think most of that would be scaling pilot to demo and not necessarily large scale commercial. But there are a lot of products that can be produced at demo scale for the first 2 years of their life before they need to really go into much larger scale. It's just coming up with the business plan and strategy, and thinking of your business as a business and not a giant science experiment, because you got to move as you're moving into demonstration. You suddenly got to shift a lot of resources from the engineering and microbiology to the business development and the client interactions. And the two areas that I'm most familiar with from 14 years in the food ingredient industry is the formulation and application work. When you have an ingredient going into a branded product, if it's a food product, as an example, that's a hot area right now. You've got to have the right texture in that product, got to have the right flavor profile. You've got to have the right shelf stability. And it's multiple ingredients that all get commissioned, going through taste testing to find out from a sensory standpoint, does that hamburger taste similar, almost identical to a full beef hamburger? If you're making milk or an egg replacer, is this texture, is its sensory attributes fulfilling? And that in itself is a science. I mean, there's scientists that are specialized in flavor chemistry and in texture chemistry and the analytical tools to measure texture. When I was brings back memories from my days, 11 years at Opta Food Ingredients. We were on the leading edge on developing, novel starches that you couldn't tell the difference between full fat sour cream and our fully non-fat sour cream. It even the texture right down to it would melt on a baked potato just like a full fat sour cream. That was material science on steroids, let me tell you, because I was involved in that. Was one of my first and only patents was that particular product. And it was designed not at the bench, it was designed in the pilot plant, because what we were doing was thermochemistry with the starches to have them behave in the way that they would behave. So it's pretty interesting stuff back then. Still interesting today, which sort of gets me, I get pulled back more into the food industry from the biotech side now than what I would have ever imagined, because my understanding of the food space.
Yeah, it's been really interesting, isn't it, to see the pivot over the last 5 years into the food industry. And I suppose half the challenge is being able to kind of get the economics working, You know, I think the whole challenge is across the industrial compared to the pharma space is getting things to work in, you know, at scale at a price competitive aspect, and that seems to be like the biggest challenges firms have in you know, becoming successful.
Yeah, yeah. I mean, you're saying, I'm getting very much more involved because of some of the work I'm doing in the area of carbon fee and climate legislation, getting more and more involved in understanding the need for regenerative agriculture. Basically putting carbon back in the soil. When you look at, right under our feet, the largest sequestration method that we could undertake worldwide would be soil carbon sequestration, and it gives us two bangs for the buck. It's a means of removing carbon out of the climate to give us climate restoration, because just being net zero isn't going to get us where we need to do. We need to be taking carbon out of the atmosphere, and at the same time, in order to feed the populations that we have, we need to move towards these regenerative practices that allow us to remove soil, return soil organic matter. And what does that do? It returns the microbiome of the soil to where we don't need to over fertilize. We don't need to use the levels of chemicals and pesticides that we have. And it requires a change in the supply chain in terms of what products the chefs will be using. All these different kinds of plant and agricultural ingredients that can make much more nutritional products for the consumers, and it's just amazing. And you actually see a convergence of regenerative agriculture with synthetic biology, because guess what's happening? You have companies like Indigo Ag, Pivot Bio, Arbiom, all these companies that are working on biological solutions to put organisms that have been killed off, get those organisms back into the soil, because they have a synergistic effect with the roots of the organisms. Then they can affix nitrogen, they can deliver the nutrients that are needed more effectively to have much healthier plants. And you start rotating the plants differently. I mean, Europe's been doing things closer to regenerative agriculture, sort of indirectly in their traditional farming technique through the year, because if there's produce, it doesn't get wasted and end up in a landfill. It gets used as feedback to feed animals, and then the animals naturally graze, and like in Germany, they're having something planted on the soil. You don't see barren farmland anywhere in Germany. I don't think you see them much in the UK. Simon, when I was over there quite often with Green Biologics, I'd drive across the countryside from London out to Oxford, and I don't ever remember driving by a field and seeing it be bare soil. There was always something growing on it, which is what makes the soil healthy and makes these more healthy ingredients.
Oh, it's a great space.
Interesting times we're in.
Yeah, without doubt. And tell me just what I found quite interesting. Some of the feedstocks used within the sector's changed so much over the years as well. Now you're talking about companies using gas, kind of methane gas, or using components of kind of CO2, for instance, to use that as a particular feedstock in the process. And that must be quite an exciting kind of change and alteration within industrial biotech.
Yeah, those are some interesting. The gas fermentations, in my mind, and many other of the other engineers that I work with and know well, we always have lengthy discussions, and we always conclude that we don't think gas fermentations are going to be a long-term approach, because they require a specialized fermenter design, and the mass transfer characteristics are different. They're easy. I mean, they're interesting, but I think they're much more difficult, much more problematic than what people might realize, versus liquid fed fermentations that are aerobic or anaerobic in nature. But certainly there's other interesting feedstocks beyond just sugar. I've got a client company that we're producing a product using methanol as the feed. So you can imagine, ultimately we'll be able to produce fairly inexpensive methanol, because from anaerobic digesters, which are producing methane, you can convert that methane into methanol and then have a methanol fed fermentation to produce protein types of products. Glycerol, a byproduct of biodiesel plants and a byproduct of ethanol plants, is a great carbon source for feedstocks. So there's a multitude of different avenues that folks are looking in. I think, when you look at large scale fermentations and look at some of the limitations of going large, I'm thinking more and more in terms of oxygen, using liquid oxygen and being able to boost the amount of delivery of oxygen to an organism by direct oxygen injection. The cost have come down so much on the technology for hydrogen, nitrogen, as well as oxygen and CO2 separations, that it can probably, it's to a point where you've got the trade-off. You can have an increase in OPEX because you're going to be injecting oxygen to get that high oxygen uptake rate, versus the high investment in CapEx to make this mammoth fermenter with a mammoth agitator on it and large amounts of compressor horsepower to deliver gas to the fermenter, where maybe you look at the trade-off of that CapEx and the operating costs for the amount of agitation and compressor horsepower, maybe oxygen injection, just at certain geographical locations where it's available, might very well make sense.
Great. Well, thank you for that. And what about in terms of the right partners? A load of smaller, earlier stage kind of companies that are dealing with these scale-up challenges, for instance. Well, what's the best way, or who are the right partners for them to help, to help them scale up their processes?
I think there's a number of engineering firms and subject matter expertise kind of people out there. I personally made the choice to get involved with Lee Enterprises Consulting. Lee Enterprises Consulting, we're all independent consultants, but our common thread is we're all members of this Lee Enterprise Consulting network, which is the largest network of subject matter experts that is focused on the bioeconomy and solving issues within the whole bioeconomy sector, whether it be fermentations, it could be biomass conversion, it could be pyrolysis. And we work in every aspect. We're just not a bunch of engineers. Many of us are ex-executives. We do financial modeling. We work with investment groups on doing due diligence on technology. So we have a team of people that can work on due diligence. So we can deploy teams of people to help that small company or mid-sized company figure out, help them develop their strategy. That's an area I'm involved in specifically. I do a lot of work with CEOs and CTOs, just helping them figure out strategically their plan going forward on how to commercialize. How long is it really going to take? How much money is it going to really take to do that? And start spending frugally early before, as I tell people, you don't want to go out and do a big money raise and then spend it like drunken sailors. And 5 years down the road, you go, wow, we wish we wouldn't have hired all that direct headcount. Well, you don't have to. You can bring in people like myself and others that have been around the block, that have built things and know we've got the lessons learned, and we're very willing to share those lessons learned to help people develop their strategies and help them develop their plan forward on how to scale up, as some people say, or in my terms, I scale down. So having people like Lee Enterprises, and there's others out there. It just so happens we've got roughly 150 subject matter experts that can across the board. We can handle food products. We can handle environmental, issues. When you're siting a plant, there's a lot that goes into, understanding where you might site a plant based upon its geographical location and impact on the environment. Workforce is really key, in terms of locating geographically where you can find a good workforce. So we're the kind of folks that have those lessons learned, that know what mistakes that you don't want to take that could be fatal errors, because you don't want to have a fatal error. You can make a minor mistake, that's fine. You learn from those, but you don't want a fatal error to occur.
Absolutely. Probably pays to pay for a bit of gray hair in the space.
Yes, quite often. I was fortunate enough, I learned that early on. I had some great mentors when I was younger that were guys that were like at Ashland Chemical, Newt Farrell, God bless him, I'll never forget. He was probably 3 or 4 years from retirement. He taught me so much about what really happens in chemical engineering. And I worked with him to help build the Ashland Chemical pilot plant in Dublin, Ohio. What an experience, as a co-op student, and then right out of school working with people that had commercialized processes and knew how difficult it is to commercialize. And you don't learn that in school.
No, exactly.
So those are some of the early on, molding and shaping things that really got me even more interested in what it takes to build commercial plants. And it's a lot of work, but I'll tell you, having been involved in startup after startup my whole career, for me, it's so much fun. And I've probably worked on almost 20 startups, either starting from scratch or a new business unit within an existing company. They've every one has been really fun.
Well, that's great.
And I'm still doing it.
Yeah, no, that's
with my consulting business.
Well, look, Joel, that's wonderful. Thanks so much for coming on, sharing your story and sharing your kind of expertise. So really appreciated it. Great to have you on.
Well, thank you, Simon. It was it's been a pleasure. You had some good questions, and hopefully, some of what I've had to share with you today, some of the younger startup companies, people that are interested in the space will learn more. And I'm always available to come back on or to talk to anyone out there who's looking for help. I want to see synthetic biology and industrial biotech be successful in the reinvention of the chemical industry and now the reinvention of the food industry. It's exciting times.
Absolutely.
Thank you.